Hi, I'm Emily DeRosa. I'm a recruiting specialist here at Northeastern University.
Hi, I'm Srirupa Chakraborty. I'm an assistant professor at Northeastern University in the Department of Chemical Engineering and also jointly appointed in the Department of Chemistry and Chemical Biology.
And this is the Faculty 5. What types of research are these students participating in, and how are you interacting with them during that research?
So let me tell you a little bit about the type of research our lab does, and those are the type of research the students are involved in. So my research interest is in applying computers to solve biomedical problems and at the molecular level. So we use computational modeling to understand and engineer complex biomolecular systems. So these are the type of problems that we look into, especially the problems that are not easily solvable by experimental methods. That's where computers come to the rescue at a very small scale, at a very small time scale. And there are a number of different problems a number of different biomedical challenges that are being addressed in our lab. We've got, problems dealing with trying to predict better antibody-antigen interactions. We are looking into trying to design better biomaterials inspired from nature. And different students are working on a number of such different aspects, all towards the goal of using computers to solve biomedical problems.
What is your favorite part about the research that you do? What really drives you to do the research that you do?
I think the biggest part, and sometimes it's also the biggest challenge, is the uncertainty that's there in research. There's uncertainty, there's unpredictability, there's sometimes setbacks. So you expect a certain answer to your research and then things don't pan out the way they are. However, it's like solving a puzzle, and you keep at it, you try to analyze the new data or the unexpected data, you discuss with your collaborators, and ultimately when that last piece of puzzle falls into place, the feeling is magical.
How long have you been researching this topic, and how has the research changed over the years?
I've been interested in application of computers right from, I would say, middle school. But it was during my undergrad days that I started reading about, the small scale, the nanoscale. I started reading about Richard Feynman and his call for exploring, his challenge for exploring. One of his quotes that I really like to say is that everything that living things do can be understood in terms of the jigglings and wigglings of atoms. So it's these jigglings and wigglings of atoms that we are interested in. You remember I mentioned that we work with complex biomolecules. Now, when one mentions biomolecules, the first picture that comes to mind could be images that we see in our books, right? We've got images of DNA, RNA, proteins. However, these are not just static objects. These are extremely dynamic, and it's their motion, it's these jigglings and wigglings that ultimately result in their functioning. It ultimately gives these molecules their personality. And with computers, you can look at such small scales in the nanometer range scales and the angstrom range scales, and in time scales of femtoseconds to nanoseconds and microseconds, which is not easily accessible by experiments. And that's what we as computational simulation and modeling scientists can achieve. And I think in this regard, it's improved a whole lot over the years. And nowadays, we frequently do simulations of 1 million atoms, going up to 70 to 100 nanoseconds per day even. So it's ramping up very fast. It's now the era of data sciences. It's now the era of computer sciences. And I think chemical engineering and biochemical engineering is doing a good job of keeping up with it.
What would you say are some of the biggest findings or most surprising findings that you've had in your research?
So very specifically, the biomolecule of interest that we have is looking at something called glycoproteins, or proteins and other large molecules that are covered with sugar, so literally sugar coated. So one aspect of that I was initially not aware of was that a lot of these viruses, even SARS, that we unfortunately are also familiar with, but I began my work on HIV, the main viral protein to which the antibodies bind have a covering of sugar. So they are literally sugar coated, and they camouflage themselves with these sugars, and antibodies cannot bind as effectively, more so for HIV, and that's why we do not have an effective HIV vaccine. And it's very difficult to study these glycans experimentally, and for a long time, for decades, these glycans were completely ignored. Even for crystallizing these structures, to know more about their structures, the glycans were just removed. But ultimately, it was found out that they play such a critical role because they form a barrier to the antibodies binding. And going ahead using our computational methods to better design, to be able to better capture the structures and dynamics of these glycans, we've always been keeping on getting surprised as to the impact of these glycans and how wrong the community was in ignoring these glycans. But I think everybody's keeping up. It's being an upcoming field where more and more methods are being developed, both in experiments and in computation, for studying these glycans, and more so going forward, I think. So we are working on that too, trying to get inspiration, get ideas from how nature has designed such glycoproteins in solving other material problems.
Thank you so much for taking the time to chat with me today.
Thank you so much. It was lovely talking to you, and thank you for having me here today, Emily.